Hello all. In the unit Chromosomes and Cell Division, let us look into the topic Euchromatin and Heterochromatin. First of all, let us know what is a chromatin. A chromatin is a complex of two things. One is DNA and the other is proteins. Here the proteins are particularly histones. They appear like beads on a string. This is a chromosome which, when extended, appears to be like a thread-like structure, and this is called as chromatin. These green color structures, they are the proteins around which the DNA is bound. Now the chromatin can be further classified into euchromatin and heterochromatin. This is a cell which has a nucleus. A cross section of this shows the interface chromatin. Here the single chromosome appears to have lighter regions and the darker regions or the darker bands. The lighter regions are called as euchromatin and the dark regions are called as heterochromatin. When this chromosome is extended, we can see the appearance of heterochromatin and euchromatin regions. The heterochromatin shows the densely packed regions, whereas the euchromatin regions shows the loosely packed regions. Let us now look into euchromatin. The euchromatins are well dispersed form of chromatin. They take up the lighter DNA stain. In this picture, you can visualize that there are two regions which are lighter as well as darker regions. The lighter ones are the euchromatins and they are genetically active. This is the most important thing in euchromatins. So how are they genetically active? That is, they are involved in gene duplication, gene transcription, that is DNA dependent RNA synthesis and phenogenesis, or otherwise phenotypic expression. That is, they produce a particular protein. The euchromatin, they contain large amount of DNA as well as the structural genes. These structural genes are helpful in replication and transcription, and this occurs during G1 and S phase of interphase. Next is the heterochromatin. Heterochromatin are highly condensed form of chromatin and they take up dark DNA stain. This is the heterochromatin region. They are genetically inert, that means they are genetically inactive when compared to euchromatin. These heterochromatin, they are found around the nucleolus and in the periphery. This is the peripheral region and this is around the nucleolus. They exist both in centromere regions and in the sex chromatin, and they are characterized by high content of repetitive DNA sequences, but these are not transcribed. Further, the heterochromatin can be divided into constitutive heterochromatin and facultative heterochromatin. They can be distinguished based on their features. Let us look into constitutive heterochromatin. They are the stable form of heterochromatin which do not loosen up to form euchromatin-like structures. They contain repeated sequences of DNA which are called as satellite DNA, and they are found in centromeres and telomeres of chromosomes. Whereas the facultative heterochromatin, they are reversible form. They can change the structure depending on the cell cycle, and the best example are the Bohr bodies of females. And finally, the differences between heterochromatin and euchromatin. The heterochromatin are tightly packed form of DNA. They stain dark and they found in the periphery of the nucleus, particularly in the eukaryotic cells. They are generally inactive form and they are compactly coiled. Whereas the euchromatin is loosely packed form of DNA, it stains lightly and they are found inside the nucleus of both the prokaryotic and eukaryotic cells. They are genetically active and they are found to be highly coiled. So this is all about the euchromatin and heterochromatin. Thank you.